Welcome everyone, and thanks for Cure Duchenne for inviting Anna and I today to share with you some DMD clinical trial updates and overview. So I'm Lauren Morgenroth. I'm a certified genetic counselor by training, and I'm the Chief Executive Officer for TRiNDS.
And I'm Anna Christensen. I'm a lead project manager in TRiNDS Strategic Solutions. So it's great to be with you all.
So we just wanted to start off with some very quick disclosures. Anna and I are employees of TRiNDS, and Cure Duchenne is one of our partners and one of our founders. And in the next slide, I'll actually tell you a little bit more about what TRiNDS does, but for the sake of the disclosures, TRiNDS receives grants and funding from pharmaceutical companies as well as academic centers. So a little bit more about just TRiNDS so you understand who we are and where our expertise comes from. TRiNDS does stand for Therapeutic Research and Neuromuscular Disorder Solutions, and we're a specialized neuromuscular CRO that was launched in 2016 due to an unmet need for a dedicated clinical research partner that could bring meaningful and expert solutions to sponsors and academic centers for their clinical studies. So a CRO is a company that supports pharmaceutical, biotechnology, or device industries for those research study needs. And our team at TRiNDS is made up of experienced and passionate members working to deliver this support. We have leveraged our experience from the academic Synergy clinical trial network that was formed in 2000. So we have over 20 years of experience running focused neuromuscular and mostly DMD Duchenne trials. And in the last 4 years, we've successfully completed 5 very complex clinical trials, with one achieving the ultimate goal of receiving regulatory approval. We support a wide variety of projects from phase 1, and Anna will tell you what that means, through to post-market registries, as well as observational and natural history studies. And so Anna and I are going to tag team in the next kind of 15 to 20 minutes to provide you more detailed information about the clinical research process. And I'll close out and share how some of the events of the last year could be used for us to think about ways to improve or speed up that research process. So Anna?
Great. So it's great to be with you all. And so I'm going to review kind of how things work, so what a clinical trial is, and how to navigate this process of deciding whether to do a clinical trial. And then, like Lauren was saying, she's going to walk us through how things have changed and what we've learned. So, you know, when you think about getting to where your doctor can prescribe a new medication, you have this idea that somewhere a scientist comes up with an idea in a lab, something mystical happens, and then all of a sudden it's at your pharmacy. And as I'm going to walk you through, there's a lot more steps and work in the middle that you might not know about and that you will become involved with if you join a clinical trial. So here are the different phases of research. And so you'll hear a lot of presenters talk about the phases of research, that it's a phase 1 or a phase 3 or preclinical. And here's what we're talking about. So first, the idea begins in the lab. And that's where you test it before it comes to humans. So you might test it under the microscope. You might test it with animals. You're making sure that your idea works the way that you think it might. Once you have enough information, then you can move into what people call the clinic or clinical trials. So phase 1 is your first time in people, and the focus of phase 1 is safety. Once you know that it's safe enough to continue studying, then you can move into phase 2. And phase 2 is where you continue to look at safety, and you also are looking for effectiveness or efficacy, which means, does it work the way that you think it will? Then if it passes that, then you can move into phase 3, where you monitor side effects and you're really proving effectiveness. So the question is, in a larger study, does the effects that you saw way back here in the lab continue to hold true? Then hopefully you submit for regulatory approval. And this is the step where if it's approved, your regular doctor can prescribe it for you. And you move into phase 4, where you continue to study the new product and you're monitoring for long-term problems. So this is what happens in general. This doesn't matter if it's Duchenne or something else. This process happens all the time. The piece that's a little different with Duchenne is that Duchenne is a rare disease, which means you can't have studies that are quite as long or quite as large as you might hear about. So everyone has heard about the COVID-19 vaccine trials enrolling thousands of people, and there just aren't that many Duchenne patients who would qualify. So in a phase 1 in Duchenne, you might have a smaller phase 1. It might be, this says 20 to 80, but in Duchenne, it's often less. Phase 2 might be still small. Here it shows 150 for your typical study. This might be a lot smaller than that. And a phase 3, a very large phase 3 for Duchenne, is around 200 participants. The other thing that you can hear about is sometimes one study will really take up 2 phases. So you might hear about a phase 1-2 or a phase 2-3. And what that means is they're really trying to answer the multiple types of questions in the same study. And so that's pretty common in Duchenne muscular dystrophy. But this just gives you a lay of the land, that it goes from the lab, from an idea to the lab to clinic in 3 phases. Then if it gets approved, you move to post-marketing research or research about an approved product. The most important thing to know is that a clinical trial is an experiment which answers a question. And it's, what does that mean? And the most important thing for families to know is it means we don't know the answer. We're doing the trial to answer the question, but we don't know how it's going to turn out. Everyone is hopeful that you'll find something new, but we don't know for sure. And it also means that even if you're taking an investigational drug or product in a trial, it's really not the same thing as your medical care. The purpose for all the things you'll be doing in a clinical trial and all the tests and all the doctor's visit is really around answering a question. It's really not about providing the highest quality medical care that your usual Duchenne clinic might do. So I think it's really important for families to know that even though the things you do in a clinical trial and the people you see might be really similar to your Duchenne clinic, it's really not the same thing as clinical care at all. All right, so we're going to run through some terms because you will hear a lot of these terms through all the Cure Duchenne and other workshops that you might participate in. And so the first one is inclusion and exclusion criteria, which is a very fancy way of saying rules about who can and cannot do a study. And in general, you're designing these criteria or you're designing the rules so that you're finding people who are the most likely to benefit and the least likely to experience risks, because the purpose of a trial is to answer a question, and you don't want to put people at risk just to answer a question. The next piece is you want to find people who can do the tests and who can measure for whom the test will show a real effect if there is one. Endpoints. So that is the result that shows whether the study intervention is effective. And usually it's a parameter that you analyze. So your endpoint might be muscle strength. And the way you get to the endpoint is by analyzing the results of a test from baseline. Outcome measures are the tests that help you measure the impact. And so studies may have multiple outcomes that all feed into answering a primary endpoint. Schedule of events is a big table or map that shows when, where, and how the tests are done. And most studies will have these, and we will keep going here. So one question that everyone has is, why the heck are these trials so strict? And the reason is we have multiple different kinds of rules that cover the activities that we do in clinical trials. So first and foremost, there's a lot of rules about safety. And these rules are set by agencies like the Food and Drug Administration and other regulatory agencies. They're also set by the institutional review boards. And these are rules around how do you keep people safe when they are participating in a clinical trial. So how do you keep them safe as possible? And it has to do with how problems are handled, how your study is designed, who can do the study, who can't do the study. That's how those safety rules come into play. So the other bucket of rules is rules about good data. So if the whole point of doing a clinical trial is to answer a question using data, you want to make sure that your data will be able to help you. So, if you collect your data improperly or if you have mistakes, then you're not going to be able to answer your question. So there's tons of rules in the protocol that will be all about how do you collect data consistently? How do you do it the same way every time? Last, there are rules to protect your rights. And so these are safety and ethical rules that are set by the institutional review boards. These are ethical groups or IRBs that are making sure that everyone participating in a study is there because they want to be there and because they've made an informed decision. So how does this all come together? So first, a researcher makes a protocol which describes everything they're going to do and what questions you're going to answer. That goes to the regulatory agencies like the FDA and IRB. Everyone in their review makes sure that the protocol is following all the rules and gets everyone approved. But this protocol is like a promise. It's a promise you make to the regulators and the IRB, and you're required, if you're a researcher, you're required to follow the rules and the protocol exactly as they're written. So, which brings us to the next question is, why can't they change the rule just for me? And the reason is that you really are very strongly discouraged from making changes because that protocol is a promise. And here's how that plays out. Here's why that's important. So for example, if I am somebody who has a diagnosis that's not allowed in the inclusion-exclusion criteria. So let's say the study excludes me if I have severe asthma, and I have severe asthma. Here's how it might play out is that I might have a side effect more often, or I might have a new kind of side effect. But at the end, it will be very difficult to tell if this side effect is due to the drug or if it's due to the fact that I have asthma. If I'm somebody who can't do a study test well enough, so maybe I can't walk far enough or I can't move my arms well enough, what it means is I might have an improvement in my symptoms, but my study test can't measure the improvement. And so then at the end of the study, if my scores don't change, we have no way to know if it's because the drug didn't work or because I couldn't do the test at the beginning. Similarly, let's say I do the study test too well. So I can walk too fast or I don't have enough symptoms. And I might still have an improvement in symptoms, but I might not be able to measure it. And it's because I score 100% at the beginning and I score 100% at the end. Maybe my symptoms really did get better, but the test couldn't pick it up. So what this means is at the end, we might not know, does the fact that my scores didn't change, is it because the drug didn't work or because I was already at 100%? So this is the reason why very often researchers are not able to change the rules to allow individual people to enroll in a study if they're not eligible. So there's a lot of steps in getting a research study going. And what I wanted to highlight is that it takes a fair amount of time. And the funny thing is, you might have long periods where you don't hear anything. And so on this slide in the green boxes are things you'll hear about from the community. So what you'll see is it'll seem like nothing is happening for a long time. And then all of a sudden you'll hear that a new study is approved by the ethical or other regulatory approvals to do the study. And then people will start looking for the patients and people will start signing up. And then people will finish the study and it'll feel like nothing is happening for a long time. And then all of a sudden you'll hear the company or the researchers presenting the research results. And then again, it feels like nothing is happening. And then all of a sudden either the drug is approved or not approved or a new study is open. And it feels like this comes out of nowhere. But what happens is first we design the protocol that we talked about. Then you choose your investigators. You figure out where you're going to do the study. And then you get the approvals and people start signing up. And then as you go, the data gets entered and cleaned and reviewed. And then the statisticians analyze it. And that takes a long time. And then we start sharing results. Then you start submitting to regulatory agencies to share the results. So you made the promise of the protocol, and here's where you turn in your work. And then as a result, the regulators will make decisions about whether you should do a new study, if the drug is approved, if the drug isn't approved, all kinds of things will happen there. So this is what's happening behind the scenes before you get started with a study. So last, I'm going to walk you through informed consent. So informed consent is a process. It's a discussion. It's how you get to where you are making a decision where you understand what's being asked of you. You've had a chance to ask questions. You've had a chance to think about whether or not the risks and benefits work for you in your particular situation. So it's more than just forms that you sign. So there's a lot of different ways you can do informed consent. So you can do informed consent by in person. You can do it online. You can do it by mail. Some studies will, for outside of clinical trials, will do it by phone. But no matter how you do it, there will be the opportunity for you to ask questions. And it's really important. But there's more than one person that you could ask these questions to during this process. So first, the lead researcher at your site is called the principal investigator, or site PI. They're the doctor in charge of your particular site. And they can really help you with questions about what the trial is about and risks and possible benefits, especially things that were seen in preclinical research or earlier trials or the literature. The person who works for the PI is the coordinator, and they are the person who will be scheduling your visits. They will probably work with you at a visit. They might be involved in getting the paperwork organized. And this is the person who can really walk you through how the study visits work and how it might interact with your routine Duchenne care or your life outside the study. And your regular Duchenne doctor is still really important. So sometimes your site PI will be the same person, but if they're not, it's really important to stay in contact with your Duchenne doctor because they can walk you through how the risks and benefits that the site PI told you about might affect you personally, given your own medical history. And they can also help you navigate how to or whether to change your routine care outside the study. Finally, it's really important to remember you have resources like Cure Duchenne, and advocacy groups can really help you with understanding what the trial is about. They often have resources like this that can help you understand all the concepts, and they can help you understand what this would mean for your family if you sign up for a research study. So I think the important thing is to remember that you have more people on your team who can help you understand what's going on and make a decision that you feel okay about. So next, I'm going to hand it over to Lauren because this has been an exciting year, and she can share with you how the last year or so has affected how we do clinical trials. Lauren?
Thanks, Anna, for going through all of those important processes, and I'll touch on a few of them just in this first slide here. And so as Anna said, I really wanted to take this opportunity, and it's hard for us to kind of not talk about what's happened in the last 12 months with this global pandemic. And so just to walk you through these final slides, I'm going to just go through it by thinking about the 5 W's. So the why, the what, the when, the where, and the who. And so this is the why. Obviously, I think we all know why we are here, talking about kind of things that can improve on the research process. And what I've done here is just wanted to show and overlap 2 different timelines. The one on the bottom is the one that very often is seen in your typical drug development, a discovery process, where Anna's talked to you a little bit about those kind of different phases. Oftentimes, just that little slide she talked to you about of the clinical trial can take anywhere between 6 to 7 years. And then if you count the time that it takes to do some of that preclinical, it can vary and be, 3 to 6 years. And so that process is typically taking 10 to 15 years. But we've now seen that in 12 months, we were able to get an identification of a new disease, get multiple types of drugs, vaccines through the process, through multiple clinical trials, large clinical trials, to ultimately get not 1, not 2, but multiple drugs approved for vaccines and other indications. Granted, they're emergency approvals, but I think we would all take that with hopes that more data can be collected to confirm and get full approval. And so really kind of what I want to highlight here is that I think this has been a great reflection of a year to say, well, we got to be able to do things quicker if we were able to do them. And maybe there's ways we can improve on it. So on the next slide, I wanted to think about just what are some of the aspects that we could maybe improve upon, speed up that process, and think about ways of being even more patient-centric because of all the burdens we really kind of fully saw occur to everyone worldwide in the last 12 months. And so first and foremost, I'd be remiss to not be obvious of do no harm. And kind of any type of modification, or when we think about adapting, we want to just make sure that the integrity and the safety of the trial participants remains truly intact, and that both the anticipated and the unanticipated harm can be identified and managed. So this is exactly why some of the regulators quickly, changed things and adapted, issued a bunch of guidances to help some of the companies and the sponsor change things, because we were changing where things were happening, how things were happening, and it was going at a really fast pace, much faster than we were typically accustomed to. It will often also come down to, again, that risk benefit of thinking about kind of what types of trial modifications can we do that can, make it go quicker, but still answer the ultimate question. Are there ways that we can, adapt or modify the access to the trial intervention? What are some of the impacts of moving the actual location? So thinking about having more things be potentially done at home, and could that be causing, undue bias? And then what happens if we actually start thinking about alternative controls and making more use of that real world data? So in the next slide, I'm just going to go through 3 case studies quickly of what TRiNDS has done in the last year to kind of pivot, as many of you have had to do. And so the first one is the actual administration of the drug. And so, for many people, they think, well, duh, we should have been doing this from the beginning. But I think, again, this is not something that kind of inherently is thought of, because we have these face-to-face visits. This is the opportunity to educate on how the drug's being given, following the returns of medication. But we really saw that in some cases we could have the medication shipped directly to the home. So wanted to just make that case of, considerations early on of just saying, is it safe to be able to do it? And are there methods for being able to still control the return, making sure empty bottles are still returned? That's still very important to demonstrate that the medication has been used and taken properly. So there are kind of a whole slew of things that happen a bit behind the scenes that you will then interface with if you have to start having, pre-shipped labels, boxes, so all of these things can be sent to the home. The second scenario with this kind of administration of the drug, in some cases, drugs need to require an allied health or a nurse to come to the home to potentially administer the drug. So those were, again, ways we wanted to really adapt. I think there the take-home message is really making sure these nurses are trained with pediatrics. We've seen that in times of COVID, they were just trying to send any type of nurse, and even of periods of non-COVID. So it is really critical that we obviously work to find very highly qualified nurses that know that these patients are going to be hard sticks, and have that pediatric training. And then the third, just kind of quick case series that I wanted to highlight was about the location of where these assessments take place. And this sometimes can be a little bit tough, because you have to really require some careful considerations of prioritizing which of your study endpoints can be done at home versus in the clinic, and how that's actually, again, controlled and monitored and reviewed. So there's some, critical endpoints that regulators like to see that require courses, where you need to have a kind of measured 25-meter course or a 10-meter course. Those may be harder to do in a home setting because you don't have that standardized measurement of that course or stairs or other things. So it is, again, something that we are considering of finding ways to do some assessments at home to, again, avoid that burden of travel, which gets me to where. So this is our third W, and I wanted to start off the where with a quick fact, so that 70% of trial participants live greater than 2 hours from the nearest study site. And so I would actually guess that in Duchenne, that's probably very conservative, and especially for trials that are specific in terms of gene therapy or disease modifying interventions that often only have a handful of sites, I would probably say that's even higher. And so what I wanted to kind of point out here with the where is, where are we actually making these modifications? And so I think really in all components of the research process that you heard from Anna. So we want to kind of think about ways we can recruit more efficiently, remotely, maybe start using e-consents where you can have the conversation with the doctor remotely. We've seen that e-telehealth and e-vits were happening quite well for the most part. I think we still need to reach those that don't have those access to computers. And then also ultimately we want to look at ways we can do virtual visits, digital health, ePros, all of these things here. And ultimately the take-home message here is really about the technology, and we want to just make sure that we use it to empower us and not control us. And so the last one that I wanted to get to was the who. And so who are the individuals that we need to do this? And so it will be kind of a collaborative effort to be able to drive this, and all stakeholders play a key role. So the sponsors need to participate, and they need to have an ability to generate reliable and valid evidence. We need to have the regulators to review and trust the data in a more timely and efficient way. We need all of our investigators and our site staff to kind of help execute these trials in a safe and nurturing environment, and really kind of think about some of these technologies we can do to really help be efficient. The trial participants, we need to make sure that things are more flexible and access is more readily available, and that you have more control over your experience in these studies. And then technology and service providers, we need to also be prepared to empower all of these stakeholders to include them to make the trials more efficient and faster without causing greater burden or difficulties in an environment that has a lot of moving parts, as you heard. And so with that, we wanted to just thank you for your attention and listening, and we're happy to have you submit any questions to our info@TRiNDS if you have just gaps in some of this information or want us to help you work your way through this process. Anna?
Thanks, everyone. And you can always get in touch with the Cure Duchenne team if you have any questions. But thank you so much. It's been a pleasure joining you today to talk about clinical trials.